24 to 36 hours, like I showed you in the Western blot. So what we think is Zapshot is like a mechanism to clear off those interferons. Because I told you, chronic interferons are bad for the host. It could cause autoimmunity or tissue damage. So we have a system where one gene is an antiviral protein, the other gene, other isoform goes and clears up the interferon once the viral infection is over, right? Which is really important. The negative regulators are really important in balancing our immune system. But what we are thinking is maybe there are viruses which can modulate splicing and make Zapshot very early during infection. And maybe that will help chopping up the interferon so that the viruses can live in. So as you can see, this is a perfect example of the host pathogen battle in our bodies happening every day to protect us, right? So the big picture I want you to see here is that viruses hide from our immune system, and they hide in very intriguing places, right? In our own, using our own cellular membranes. And the job of interferons and interferon-induced ISGs are to figure out how to get into these places and neutralize this virus. And I've given you examples of the viruses which use this system. And as you can see, it is not only those viruses, but there are plenty of other viruses. And I've highlighted in red is SARS-CoV-2 as well. And MERS uses this, SARS-CoV and MERS, and I know SARS-CoV-2 as well, uses these membranes to replicate or hide their RNA, hide their viral particles. So currently, we are trying to figure out, and we have a couple of hits to figure out that go into these regions where the viruses are replicating, even in SARS-CoV-2, and try to neutralize these viruses in those regions. So what do viruses do? Viruses try to block interferon, right? So that these ISGs are not made. So the question there is, what are those inhibitors? How can we better use interferons early in infection? Can we use interferons? Because there are a lot of toxicity that needs to be taken care of as well. So the take-home message is, our DNA is constantly evolving with the pathogens. And any nucleotide change in DNA can be important to ward off particular diseases. So my lab and other labs have been involved in identifying the specific changes in the DNA and hopefully leading to new therapies. I will stop here, and I will stop at a slide which Dr. Karnasagar always has previously mentioned. I'm organizing this meeting, which is a big meeting for cytokines and interferons. The world leaders come to this, and here we'll talk about what are the current concepts in SARS-CoV-2, what are the clinical trials happening, how cytokines are important, how interferons are important. This is a good meeting for students as well, because this the charge for registering, becoming a member on cytokines is nominal compared to other meetings. So you should all look into it. And I will stop there, and I hope you enjoyed this lecture, and I hope I didn't go into too much of the weeds, and I'll take any questions you have. Thank you.
Thank you, Ram. It was very, very informative and very basic to many of the people who may be listening to this video. And you gave a glimpse, yes, of a lot of the steps of basic immunology. We have friends from the US who have all been online talking to us about the presentation and sending messages. I know we don't have too much of time for questions, but I was just wondering whether all your work with zinc and zinc fingers is related to now zinc being included as one of the treatment regimes during SARS-CoV-2. And though I heard zinc long and zinc short, what is your take on
So,
the infusion of zinc with the vitamins that are being administered as a treatment regime?
right. I don't want to imply that one or the other factors are important, but I know there are a lot of data coming out on zinc, which is important. And it looks like it is important for a basic immune response. So there is no, and vitamins, of course. Everyone needs to take their vitamin D, which is supposed to be important as well to boost your immune response. General immune health is important, and these supplements are shown to be important. But I have not done any research to say this versus that should be important. Yeah.
What?
Stops.
Can you stop sharing your screen so that you can be on full screen?
Okay.
Okay.
There you are.
Yeah, thank you. I was also wondering, there's some of the treatment regimes that are suggested, because these are related to some of the questions that are coming in. And why are T cells more important than antibodies against the coronavirus infection?
Right. So it's, I would suggest it is too early to say. That is one thing, because still work is going on. And there have been data suggesting you need a T cell response. You need a B cell response for antibodies. Both are equally important for immune responses against viral diseases. And the quality of the antibodies made and the quality of the T cell response is also a factor. The question is, if these viruses are modulating the T cell responses versus B cell response is a question which currently a number of labs are looking at, including it in our department. So I think it's too early to say that whether one is better than the other, but both are really important. And recently a paper came out saying people who have mild infections still have a long-lasting immune response, and it can be a T cell as well as a antibody response. So I think both are important here.
Okay, here is another question. Though we would have been happy to have people ask it themselves, but then the questions are too many and we have to restrict because of the time. Now, one of the questions is there any genetic study that can identify specific gene polymorphism in individuals that predispose to severity in COVID-19?
Great question. There's been one GWAS which is still preliminary, and there are certain genes which have come up there, but still there needs to be more patients where now, because there are antibody tests where you can find people who had mild disease versus severe disease, who survived, who did not survive, and who survived after therapeutics, and then take the DNA and do the genomics on it and find out if there are SNPs. And I'm sure, apart from, it will be a little complicated because there are comorbidities also involved in this disease, because you're more susceptible if you have a heart condition or a cancer or other conditions, or type 1D and other things. But I'm sure those genomic studies you will see something there.
Okay, thank you. How is the manifestation of cytokine storm in COVID-19 disease and its moderation by use of steroids? Are these related, interlinked, if any? This is a question.
Right. So what is happening in COVID, based on the literature available, is initially the interferon responses are not there. So somehow the virus is blocking that interferon response and the ISG response. So what happens is if you have a condition where the viruses are replicating and there is cell death and all that, and what's happening is the body is telling, look, I'm in stress, at least the lung. So you're getting all these inflammatory cells there. So there is a lot of inflammation going on, and that inflammation usually is at one point of time is hard to clear off until you clear the virus. So using immunosuppressants has been tried, and people are using it. Clinically, I'm not sure how responsive they are, but clearing the infection is of prime importance, and there are several clinical trials looking at blocking certain cytokines to see if they're efficacious against SARS-CoV-2.
Okay. Because we have clinicians who are, of course, looking to applying some of the knowledge that they already have. These questions are related to how they are managing the cases. So here is a question which says, as cytokine storm plays key role in the manifestation of COVID-19 and the bad outcome, do you suggest any guidelines for anti-inflammatory therapies for prognosis and recovery?
Yeah, I would not be able to give you some guidelines because I'm sure there are good CDC guidelines and clinical. I'm sure, like the person said, there some of these cytokine as markers would be really important to see, like IL-6 and other things, where which could be important to know the extent of inflammation.
Exactly. I was in fact coming to that. Now this tocilizumab, which is of course this recombinant humanized IL-6 receptor antagonist. Now this interferes with the IL-6 binding the receptor and blocks the signaling. This is my question. I was just wondering, maybe there are differences between people and the way they are going to use the different kinds of strategies. Block these interferon receptors. But there seems to be, of course, quite a few options that people use. One is, of course, this IL-6 receptor antagonist. Then we've also heard about the cyclooxygenase inhibitors and anti-tumor necrosis factor. Now, in your reading papers or the clinical outcomes that sometimes are mentioned in articles, at what stage do you think each of them might be useful?
And that's a very hard questions, I think, and those are more very relevant questions, right? Because it depends on what stage the patient is. Is it full-blown virus, right? Viral infection, or they're in the earlier stages? Can we control the inflammation early on or later on? And this is the same question they're asking with interferons as well. Or can we give the interferons before, like when they're in the mild stage? What will happen if you give interferons later on? And I think clinical trials, which are ongoing all over the world, will be best to tell us, and they are still preliminary at this point of time.
If the cytokine storm is to be prevented, not the cytokines, but the cytokine storm, we find that the recommendation on the use of dexamethasone, which is, advocated. In your opinion, I know you are not a clinician, but you are working with them. So do you think the use of that steroid might prevent the storm? Because we see quite some success when we do clinicians on the use of dexamethasone.
No, like I said, clinicians would know better. And I've heard anecdotes of that, but I should defer that to clinicians. Because the worst thing for a scientist to do is to tell something which needs to be rectified or studied.
Because you have done a lot of your experiments, but have you looked at the role of oxygen? Because cytokine storm starts in the lungs, which is why the lungs get affected initially, And then as the storm loses control and starts attacking other organs, there is multi-organ failure. But oxygen starts decreasing in the body and it starts working inefficiently, including with the immune system.
Yeah.
So, have you been creating conditions of hypoxia to also understand how some of these basics
Yeah.
work in the
Oh,
body?
no, that's a great question, and we have not done anything about it, and I'm sure there. So the difference in where the field stands at this point of time is you're infecting cells in a dish, right? These are BO3 level pathogens. So having a system like that, controlling those, would surely come up later on. But at this point of time, it's because the virus has been there for a couple of half a year now. Things are starting up. The advances are going on. So maybe those things would, in time, be known. But that's a great question on the oxygen.
Because I have always wondered from the time the infection is detected, or they insist that using pulse oximeter, the oxygen reading has to be monitored. Even from the very beginning of, knowing that an infection is just set in or setting in, or in the very early stages, they watch the oxygen, and therefore it would be in the fitness of things to look at all what is seen in the laboratory at oxygen levels
Yeah.
and the depletion resulting in the damage that is brought about. And there is, of course, a question that has just come in. What are your thoughts on the idea of immune boosting?
Yeah. Yeah.
Others have asked, you know.
Yeah. It's a fair question. What I would say, and this is, if you're worried about SARS-CoV-2 now, right? What I would say, the best, until a vaccine comes out, the best way to prevent is social distancing with a mask and washing your hands for 20 seconds with soap and washing it with water is the best prevention at this point of time until the vaccines come. And I'm sure all these other things, and yes, of course, people taking vitamins, there are so many things which are being told and splashed around. But I would encourage people to have that social distancing as the primary thing. And then as the science progresses, there will be better ways. We understand this virus better.
From the University of California, we had Dr. Gandhi speaking yesterday on the use of mask, yes, but how that quality of mask which is being used by the public probably is serving also as a kind of a mechanism by which there is a certain amount of virus entry, small numbers, which is allowing an individual to develop immunity. So yesterday she was there, Dr. Monica Gandhi, and she was being extensively interviewed, and she was talking about it. She's from California, I think university there. And is there any take in the US?
I've not heard about that. The one thing I do is, like you are doing, wear masks. I think it is protecting us. And those studies are, if they are there, it needs to be replicated and reevaluated. So I would suggest the best way to protect yourself is washing your hands and wearing a mask.
Yeah, okay. I think there are a large number of questions, but since we're running out of time, I will see that people put it in the chat box, and if we can come back to you with them at a later date, we would be very grateful for you to answer them. So finally, it's my pleasure to thank you for the excellent presentation, very basic studies, but very much needed for application to look at some long-term treatments. It might look very basic to many clinicians who are today also listening to it, but we need all of these basic studies to strike upon appropriate method of treatment in the long run. And it cannot be that we are resorting to any short cuts to understand. And therefore, whatever you've presented, we are very grateful, and you've taken your own lessons from other viruses, other forms of immune system in the lower vertebrates, to understand also what's happening in the higher vertebrates. Thank you so much, and we hope this will be the beginning of a series of seminars that we can request you. I'm also taking this opportunity to thank Professor Harsha, who has always encouraged good science and loves to interact, and you would have already, in the few minutes that he spoke, understood his thoughts, and that Nitte is very lucky to have people like him at the helm of affairs to help all the teaching and research go on excellently. In fact, I don't need to thank some of our own home people, but Karuna Sagar, yes, he has always been a good support. And Anirban, who is now leading the Nitte University Center for Science, Education and Research, and who is your good friend, has been, in fact, instrumental in organizing the whole thing with his band of people. And I don't need to thank him because it's your home affair. You only can say, good, keep it up, do even better in the years to come. So Ram, thank you very much, and God bless you with great work that you can carry on, and let's hope you can collaborate with Nitte University. And yes, okay. So thank you from all of us here. Thanks a lot. Okay, bye. It's late for